Yeah, good morning. My name is Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation in Toronto. The Gairdner Foundation gives out biomedical research rewards every year to the very best international scientists, but we also want to help people understand the process of science and how science affects our everyday life. And what better in this current COVID era to talk about vaccines and how they are developed. And today I'm very lucky to be talking to Derrick Rossi, who was the founder of Moderna, which is one of the companies that has produced RNA vaccines to coronavirus. So he's going to tell us a little bit about the history. He was also a professor at Harvard Medical School, and way back when, he was a graduate student here in Toronto. So welcome, Derrick. It's great to see you again. It has been
likewise.
a while, I think.
Yeah, indeed. Yeah, great to see you. We just caught up a little bit before we started recording here. And when you told me when you retired from running Children's Hospital in 2015, well, when last I saw you, you were doing that. And it seemed like, well, maybe 2 years ago or 3 years ago, but wow, blink of an eye and 5 years goes by.
At least 5 years, and you've changed, you've moved around since then as well. So that's one of the things I want to talk to you about, is your sort of career path and what led you down different pathways. But let's go back to the beginning. Well, not the beginning. Well, yes, I guess the beginning. You are Canadian. Tell me how you were born in Canada.
Yeah. Oh, yeah. I was born in Toronto, but both my parents emigrated from Malta as teenagers. So the tiny island nation of Malta.
A large Maltese population in Toronto.
Oh, there is, yeah. And funnily enough, my parents didn't know each other in Malta, but they met in this little Maltese community in Toronto in the 1950s. Okay. And got married and had a family, and we've been Toronto-based ever since. So myself, my siblings are first-generation Canadians. I actually left Canada, though, when I was 28, and I'm now 54. So it's been a bunch of years since I've lived abroad. I lived in France, I lived in Finland, and I've lived in the US for a long time. But yeah, I mean, I did my undergrad at U of T, and then I started graduate school there. And actually, Jen, I remind you, you were actually on my committee.
Absolutely, I remember it well.
Yeah. Well, I was a young scientist there then. Let's face it. I mean, I had done an undergrad in molecular genetics at U of T, a great program. And I was always fascinated with molecular biology. I had, ever since high school. I had a really great high school teacher in Scarborough, where I went to high school, a guy named Lloyd Lumby. And he, in grade 11 biology, introduced molecular biology to the class. And I fell in love with molecular biology at that moment. It's rare that young people know their calling at such a young age. But I totally knew I was going to do molecular biology because I found it so fascinating. And then, yeah, did my undergrad at U of T, then did this graduate program, and then started traveling the planet, following, the wildest wish to fly as a young person and doing science along the way. I did a lot of science. I was at the Institut Curie in Paris for a year in a graduate program, which I did not complete. I was in Texas for a little while with an old colleague of ours, Mark Henkemeyer. Actually, I was in Mark's lab when he first started his lab. Then I went to Finland, University of Helsinki, and got into yet another graduate program. By this time, I had done quite a few years of science, so I was pretty good at it. And I was getting old enough to finally say, well, maybe it's time to finally get my PhD. So I settled in, got my PhD, published a bunch of papers. It was in the realm of, actually, to be honest, it was really from skills that I had learned in Toronto. So when I was doing my grad work in Toronto, we were knocking out genes in the lab. And it was the early day of knocking out genes. So genetically engineering mice with the intent of studying gene function in a live animal. And so I had learned those skills in Toronto, and then, used them at subsequent points in my career. But when I went to Finland, I basically knocked out all the genes that the lab was interested in studying and published a bunch of papers on those genes, which were in all kinds of aspects of biology, from early, embryonic lethality to gastrointestinal polyposis. I mean, it was really all, you never know what a gene's going to do until you knock it out. You might have a good hypothesis, but anyhow, then I turned my attention to stem cell biology, and I went to work with Irv Weissman, whom I know you know well. He's really a pioneer in the field of stem cell biology, and I learned that field quite well. And
what got you? Why stem cell biology? What sort of? I mean, you came from where it's the home of stem cell biology too. Was it?
It sure is. Yeah, no kidding.
Printed in your mind somewhere.
Yeah. Yeah. Well, I had been doing a lot of reading as a grad student, and I got very interested in, actually, of all things, aging, and realized that aging was becoming a science, a tractable science that you could actually study. And, prior, years ago, it was kind of I wouldn't call it a pseudoscience, but there was a lot of nonsense out there. But finally, you could really study it as a proper science. And I developed the hypothesis, actually, as a graduate student, that stem cells must be involved in the process of aging, tissue-specific stem cells. I mean, not, embryonic stem cells. So I had this hypothesis in my head that somehow aging must be impacting the stem cell compartment of various tissues. And when I went to Stanford, so it was just a hypothesis, and I presented this to Irv, who was the head of the lab, and he tried to talk me into working on something else, Wnt proteins and Wnt signaling. And I, because he was very interested in it at the time. And then I sat back, and it was a very fun conversation because Irv, he's a brilliant guy, but he's maybe a disorganized guy. I'm not, maybe that's not exactly accurate, but I remember sitting in his office.
He is a bit hard to describe, but I know.
Yeah, he's a giant, and he's brilliant, and I love him. But anyhow, I was meeting him in his office, and he was sitting in his chair, and I was sitting across the desk from him, but he had such a pile of books and papers. And so whenever I kind of had to talk with him, I'd sort of have to go up in the chair to get over this pile of rubble on his desk to have this discussion, which was fun. Anyhow, I presented this idea of studying stem cells and aging. And he said, really one sentence to me, he said, "Oh, that sounds like it could have therapeutic implications." And then I said, "Well, does that give me the green light to, go embark on a study?" He's like, "Absolutely, go crazy." So I then, took that, program and ran with it. And when I, for my own lab in Harvard, I basically founded the lab as a stem cell biologist, and largely aging research, but other aspects of stem cell biology as well.
Mm-hmm. So at Harvard, that's really where we get into the Moderna messenger RNA story, because you started to use that approach to reprogram cells. So I don't know the story of how you got that idea, and where, what were the bits and pieces that led to that idea? Well,
okay. So the background is that Shinya Yamanaka, again, somebody who well, had made this brilliant discovery that he could introduce 4 transcription factors into any somatic cell, human skin cell, for example, and reprogram it back to sort of developmental, early developmental stage, an embryonic stem cell-like state called induced pluripotent stem cells. We read this paper, and like the rest of the stem cell world, got, so excited by it. It's such amazing science, but I love that. I still read that paper. I love that paper. It's brilliant.
It is.
But there was a challenge. So the idea, of course, was you could take a cell from any patient, derive stem cells from that patient, and at some point in the future, and we're on that path now, differentiate those cells to a cell type of interest that the patient might need, and reintroduce it so that the person, let's say they need a liver transplant, you make a liver that is of the cells of the person, they don't reject it, it's a perfect match. That was the long-term goal of this science. Of course, the short-term goal is, disease modeling in a dish. So though the way that Yamanaka made the iPS cells then, as you know, involved retroviruses, and retroviruses are a great way to deliver cargo. He was delivering 4 transcription factors, great way to get them into cells. Problem with retroviruses is they turn into DNA, they integrate in the genome, and then they stay there forever. And these factors that he used were pretty powerful factors that you probably wouldn't want to be sort of altering the genome and leaving, c-Myc, for example, which is a potent proto-oncogene. So much of the community was coming up with ideas of deriving these iPS cells using other methods. And we had a very simple idea, which is, well, we know we want to express proteins. We want to express these, Yamanaka factors, which is really what they should be called, deservedly so. And so we thought, well, let's just avoid the DNA part altogether and try to introduce mRNA. Very, very simple idea. We know that DNA makes mRNA, makes protein, makes life. DNA makes mRNA, makes Yamanaka factors, makes iPS cells. So we thought, let's just cut the DNA out of the equation and just try mRNA. Now you have to ask yourself though, mRNA has been known to be sort of this intermediate molecule between DNA and proteins since, the 1950s. Why hadn't molecular biologists been using it in their toolbox? Because molecular biologists, whenever they can use anything in their toolbox, they do it. But mRNA was not something that people used. And sort of the short answer that most would give you is, well, it's really an unstable molecule and it's really hard to, keep it intact. It's very susceptible to degradation by proteins called RNases. And that is true, but that seemed a little unsatisfying to me. I mean, you could get rid of RNases and have a clean workspace and clean out your pipettes and I would think you could avoid that. So we made a synthetic messenger RNA. It's very easy to synthesize. It's a nucleic acid, just like DNA, very easy to synthesize. You have a DNA template. You can make a beautiful textbook mRNA, which we did. And then we introduced it into our cells in a dish, human fibroblasts. And we very effectively killed all the cells in the dish.
Oh.
Oh yeah. So that's not what we were trying to do. What we were trying to do was, express these factors. Actually, at first we were just expressing a test transcript, a green fluorescent protein from the jellyfish to see if we could get the cells to turn green. But no, we were killing all the cells. And as it turns out, the reason for that, it's amazing that viruses come into these stories all the time. We've already talked about coronavirus. We've talked a bit about retroviruses. Now this turns into a virus story because when we were introducing the mRNA to the cells, basically ever since viruses and cells first met one another, cells have been trying to keep viruses out and viruses have been trying to come up with clever ways to get in and infect cells. So cells have evolved all of these intracellular pathways that recognize invading nucleic acids. Well, what were we doing when we were putting mRNA into cells on a dish? It was basically the cell looked at it and said, that's an invading nucleic acid. Let's activate our antiviral responses. And if the antiviral response is robust enough, it leads to the altruistic suicide of the cell, because the cell is not interested in having the virus take over its control and make lots of virus and potentially threaten the whole animal. So it's a good pathway. And these, it's NF-kappa B mediated, and there's lots of toll-like receptors are involved. So that could have been the end of the story. And by the way, I'll bet you in the 60-odd years since mRNA was first described as the sort of intermediate between DNA and protein, that many labs tried to use mRNA, and many labs killed lots of cells in a dish because that's what would happen. And that really could have been the end of the story. But we thought, well, maybe there's a workaround. And we were fortunate to come across the work of two scientists at UPenn, Kettilan Kiriko and Drew Weissman, and they had published a paper a few years before that in 2005, where they were basically studying this antiviral response that cells have to injected mRNA. Or I'm sorry, not mRNA. They were actually just using short oligos, just little stretches of RNA. It wasn't actually encoding anything. They were basically trying to replicate what a viral, an RNA viral genome looked like going into the cells. And they discovered, and this is really the key discovery, that if you, so it turns out that in all cells of our body, we think of the nucleosides being in, just 4 flavors, but actually there's lots of modified nucleosides in our body. There's over 110 different modified nucleosides for mRNA. And they had discovered that if they substituted some of these modified nucleosides, when they made their short little stretches of RNA, that they could sort of abrogate this antiviral response. The antiviral, mechanisms of the cell were no longer picking up this introduced RNA as foreign and mounting this robust antiviral response. So as I said, they had published this in the setting of just, studying antiviral response and using short oligonucleotides. We read this paper and we thought maybe we could incorporate those modified nucleosides into mRNA and get it now to get into introduced mRNA, that it could become stealth, if you will, and do what we wanted it to do, which was express proteins and, in our project, reprogram cells. And so we synthesized mRNA with these modified nucleic acids, either pseudouridine instead of uridine or 5-methylcytidine instead of cytidine. And now, instead of getting platefuls of dead cells, we got platefuls of very happy live cells expressing the green fluorescent protein. And so
it must have been a eureka moment.
That's one of the classic eureka moments. Yeah, because there's only so many dead cells in a dish that you can look at, and it wears on you after a while. It's a little uninspiring to kill all the cells again. It's not our intent. So yeah, we were excited by that. Then we, went about making the Yamanaka factors. We reprogrammed cells highly efficiently. It was incredibly how efficient it worked. And then experiment I really liked in that study, we then took the iPS cells that we had made with modified RNA and then directed their differentiation to a cell type, again, using modified RNA. We used MyoD and turned the cells into multinucleated muscle cells. So it was a celebrated study when we published it because, it was very effective. It sort of got rid of this problem of bad genes in the genome, permanently altering the genome. mRNA doesn't integrate in the genome. It's totally non-integrating. But what I got really excited by, though, was the fact that every time we called them mod RNAs for short for modified mRNA, mod RNA. Every time we made a mod RNA for a new protein, and basically the whole lab started making all their favorite proteins with modified RNA because we could do it and we could get it into cells. And of course, it was cool to work with this new technology. We made dozens of proteins, and I thought to myself, we can make proteins now with mRNA. And so, DNA makes mRNA, makes protein, makes life. DNA mutation makes mRNA with mutation, makes bad protein, makes disease pathology and human disease. So I thought we could use this technology in that setting potentially, and that was sort of the genesis of the concept of founding Moderna.
Mm-hmm. Yeah, so I remember that paper. It was extremely well received at the time. As you say, other people were trying different approaches, but this again was so different and so unexpected because nobody really thought it would work. And you managed to find the sort of magic key, I guess. Do people still use it to reprogram cells?
Well, they do. So the New York Stem Cell Foundation, they've got a large array, they've called it, where they make hundreds and hundreds of iPS lines. They use the modified mRNA technology in their assembly. But since our paper came out, other non-integrative approaches have been developed, like Sendai virus. It's a non-integrating virus. And it's actually a little bit, it's easier to use. So I think and you can also reprogram blood cells, which we always had a very, very hard time. And we never actually cracked the nut of getting modified RNA into blood cells very effectively. Mm-hmm. So I think by and large, Sendai is really the approach that many use, although, and actually there's one other thing, and this is just a terrible science story, but it happens.
This is what we need students to understand, that science isn't always a straight line.
Yeah, well, we
things are wrong, but you stick with it.
Yeah. Well, and lots of reagents changed. So it's as simple as that.
Oh, okay.
Yeah, no kidding. So we published our paper, and it was, in our hands. We were just literally converting whole plates of cells into iPS cells. It was so efficacious, and it was just robust as heck, and it worked every time. We published our paper, and we were using reagents that we order from various vendors, and our media we were ordering from a company called StemGen. And the lot that we had been using for all of our studies, I guess, ran out. They were selling this media to everybody in the industry or in academia that were reprogramming cells. And they changed the lot of this media, and it was unsupportive of reprogramming.
Oh no!
There was something in the media.
Yeah.
There was something in the media that forget about reprogramming that media. So we publish our paper, everybody starts to try to replicate our results. It's a terrible story. And they're having trouble. And indeed, when we get this new media, we don't know it's the media. That's the problem. You don't know where, because it's such a complex you don't know if it's the media. There's so many steps along the way in the synthesis of these things and the treatment of the cells. We didn't know it was the media that was all of a sudden, screwing up the ability to do mRNA reprogramming. So actually StemGen, who had they didn't in-license the technology, but they had borrowed it from us, and they were reprogramming themselves and finding it robust as heck, just as we had. And then all of a sudden their reprogramming stopped as well. And then it took them about a year to track down that it was this media that was actually responsible for this. And it's just one of those. Yeah. But the timing couldn't have been worse because, everybody.
This is famous thing, and now this famous thing doesn't work. It's like, oh.
Yeah. So but there was no question of its robustness.
Yeah.
And in fact, we had a hard time publishing that paper because after the paper was accepted and it was in press, some researchers who shall go unnamed had a graduate student working on it, actually with the proper media, quite frankly. They just couldn't get it to go as it was an undergrad, actually, just a kid, and he couldn't get it to work. This person, who was kind of a powerful scientist, managed to stop our paper from being published after it was accepted and in press.
Ooh.
Yeah, it was a terrible story. And it was, kind of the first big story from my lab, not the first story we published, but, first big story. I was very proud of it. Yeah. Anyhow, we eventually.
Well, yeah. In the end, you came through, and the paper did make a big splash. But it is this. It's interesting though, because it does raise this whole issue of replicability. When it is incredibly important when you make a new discovery that everybody else can do it as well. So obviously, that was a difficult story, a difficult time for you, obviously, because it did work, it was robust, and people could get it to work, but it starts to sow those little seeds. And we've seen that. I think one of the things about the Yamanaka story was that when you think about it, when Yamanaka took 4 transcription factors and put them into cells and said, I've reprogrammed back to beginning of development, your first reaction is, can't be true.
Can't be true.
But he shared his information, and everybody, particularly the people at Harvard, immediately tried it out, and it worked. So I think that's another part about science, is that if it's a robust new discovery, then everybody will be able to do it. And that turned out to be the case for your discovery. Yeah. A little bit of problems on the way.
Yeah, there was a little hiccup in the road, but luckily it survived that. But, even Yamanaka's lab, for example, they tried to replicate our findings, and they got the crappy media, and they're like, what the heck? It doesn't work.
Yeah. But of course, we know now, that the approach works, because what you just did then to reprogram stem cells was the basis of the company Moderna, which of course has taken that technology in many different directions, including of course into vaccines. So you were associated with the company for quite a while, and then you sort of stepped away. Mm-hmm. Tell me about when you were there. Did you see what kind of directions were they looking at? And were vaccines always on the horizon? Was that an idea they had? I always kept my day job at Harvard. So I founded the company, and I was on the board of directors, and I was on the scientific advisory board, and I met with the CSO on a weekly basis, but I was never, at the working at the company. In fact, the earliest scientists at Moderna got embedded in my lab to learn the technology, to learn how to make modified mRNA. But when we were first starting out, like any biotech, thinking about bringing a totally new
therapeutic paradigm to patients and to ultimately to market to approval, we knew it was going to be a long-term effort. Well, now we're on the verge of emergency use authorization. 10 years later, the company was founded in 2010. That's actually pretty fast, quite frankly, for a new technology. Part of the challenge also was because it could have application in almost every aspect of biology that you can imagine. I mean, because proteins are involved in essentially all cellular machinations and disease pathology, and turns out they're the units of vaccines and the like. So one of the challenges was, what the heck are we going to work on? What are we going to work on first? And because when you're thinking about that, particularly with a new technology, you want to sort of set the bar as low as possible, that you can get over it as a proof of principle that it works. You don't want to set a bar up here when you're first coming with a new technology. Let's just imagine, well, if we get into this particular cell in the brain and express this rare isoform of a protein, and it really only has to be in that cell and not any other cells, because then you're all going to die. I mean, that would be a terrible first application because your technical challenge is so gigantic. So though, I will say in the 4 years I was intimately associated with the company, vaccines didn't come up a single once. And the reason for that is multifold, but largely probably mostly because of a business model. Vaccines are not a particularly great business model.
Correct. Yes.
So when you're looking at this technology that could be applied to genetic disease, rare disease, almost any other aspect of biology and medicine, and my training in the molecular genetics department at Toronto made me think of genetics and genetic disease and the basis of genetic disease, and that this could be a really good application for this technology. So we're largely thinking along those lines. But again, what was going to be the first program, that didn't evolve till even well after I left. I mean, one program that actually did gain traction was based on a study that we started in my lab. Actually, it was we made a modified mRNA for VEGF, vascular endothelial growth factor, and we made this. This is a factor that stimulates blood vessel growth and health and development. And we applied it in the setting of a mouse model of cardiac arrest. You open up the mouse chest and you ligate the vessel and you give the mouse a heart attack, and then you can release the ligation and the mouse recovers. And what we did in this study was we injected into the heart. Again, pretty high bar, by the way, for a first therapeutic, injecting this modified mRNA into the heart of these animals. And we found that the animals recovered much better than had they been injected with, just PBS, salted water. And so you would get less scarring on the heart when it regenerated. So, guess what? Blood vessel flow and vascularization is important for tissue repair. So we had started that study in my lab in probably 2011, and then it got published in Nature Biotech in 2013. And it got the attention of AstraZeneca, which is this large British biotech company, a pharma company, pharma giant, and they were captivated by that study. But they, in seeing that study, they saw the mRNA in a therapeutic application. Like I said, it's a high bar application, quite frankly. But they ended up making a deal with Moderna where they in-licensed the ability to work with 40 different modified mRNAs. So the deal that was struck was, here, you can play with 40. And it was a really big biotech deal at the time. And to be honest, Moderna had been sort of flying low under the radar in quote unquote stealth mode prior to that. And then all of a sudden there was this gigantic and when I say gigantic, financially gigantic AstraZeneca deal that was announced. And everybody was like, Moderna, what geez, we thought they were working on iPS cells. What's going on here? And that's when it was sort of revealed really what the company was, what their intent was, because it was always mRNA therapeutics, basically. So, but vaccines, if you think about it, it's actually a perfect application for the technology because it's one of these really low bar applications. You have to give a healthy individual 1 or 2 shots. You give it for an antigen, encoding for an antigen that they've never seen before, and you can easily, go into their peripheral blood, which is non-invasive, measure whether or not they've made antibodies against it. Mm-hmm. And so within, weeks, whether or not you've succeeded in doing what you're trying to succeed. That is what you call a very low therapeutic bar to get over. So it's a perfect application. And as it turns out, who knew a pandemic was coming, but well, we should have, because they come around every so often. They will come again and they will come again. But the technology is perfectly suited to respond to something like that because of the speed with which you can synthesize nucleic acid. And, you can exactly encode for the protein, in this case, the spike protein on the outer surface of the SARS-CoV-2 virus, exactly encode for that protein, have that made by the cells, mount an immune response against exactly that protein. But the speed is impressive. So Moderna got the sequence like the rest of the world when this Shanghai consortium published the sequence of SARS-CoV-2 on the web. And the next day they had an mRNA designed for the spike protein. Previous studies had shown that spike protein is definitely the thing you want to target for this family of coronaviruses. That was going to, that's the protein that makes contact with the human cell, the ACE2 receptor that initiates infection, basically. So if you could block that, you might be able to block infection. So the sequence was determined, an mRNA was made within weeks, but 42 days after that initial publication, they had a sample, a clinical grade, a GMP grade material to go into patients. Yeah. That's unprecedented in medicine. Yeah,
no, that's amazing. So I think it has been really, I think the most exciting part for me as a scientist anyway, in this whole sort of vaccine race has been the messenger RNA vaccines, because it is it's great to see a totally new technology coming into play. That, as you say, just makes so much sense when you think about it. Like, oh, okay, why don't we just do that? And let your own cells make the protein. And but people were very nervous, of course. Everybody told me, well, and it's true, of course, we've never been approved. There's been no vaccine like this ever approved before. And that's true, but the concept is really no different than a DNA based vaccine, which then makes RNA, makes protein. You're just cutting one of the steps short. And I think the regulators in the end were pretty good about it. They didn't really push particularly hard against the concept. Well, we haven't seen this before. They really just looked at the data, and the data was pretty impressive. So
yeah, well, it wasn't the first program though to go through clinical trials. So Moderna's, had different mRNAs in phase 1 and even phase 2. So there was that large body of data that was accrued that suggests that the modality itself is fundamentally safe and fundamentally achieves the goal that you're trying to apply it for. That is true. And I think that helped.
Yeah.
The more it's used, the more people get comfortable. And of course, the general public and the world doesn't know about it, but the regulators knew about these studies. So you could show this data to the regulators to say, well, look, we've made mRNA, and it's not even the first mRNA vaccine that they had. They had vaccines against Zika that had gone into clinical trials, CMV. Their CMV vaccine is already in phase 2, so that study's been going on for a while. Phase 3 coming up actually for CMV. So there was plenty of data that the regulators could, and of course they got to see all that data. And in fact, when those trials were going on, they had to see those data. So it's about making the regulators comfortable with a new modality. Yeah. And I like how you appreciate that, like, oh, it's simple. And what I like about it is, you know, it's so fundamental to the biology of our cells. Yeah. You know, mRNA makes protein. Right. And then, by the way, it's degraded. It's a really, you know, it's degraded, it goes away. Yeah. So mRNA makes protein. There's no black box there. Mm-hmm. You know, unlike small molecule medicines, which everybody takes all the time. It's, you know, there's more molecules, more medicines that are small molecule medicines than any other on the planet. Well, these are small chemical compounds that you hope they've got efficacy on what you're trying to achieve in the patient, mm-hmm. but specificity is more the issue. Yeah. Yeah. So the problem with small molecules is they interact just fine with the target that you're trying to target, but invariably they have off-target effects as well, and which can cause toxicity. And, you know, actually most small molecule medicines as they're being developed don't make it all the way to approval because they fail along the way for either an efficacy issue or a safety issue. Yeah. So, you know.
Yeah. Anyway, Moderna has been charging ahead. You left Moderna, went back to, you were always at Harvard. But since then, you've become a bit of a serial entrepreneur. So tell me about that. Did you get the taste for company creation?
Yeah, sure. Yeah. Well, I had, it's funny, I ended up teaching an undergrad course at a course that I invented, actually, because my faculty chair, Doug Melton, who you know, said, Derrick, he gave me the idea. He said, why don't you do a course on, you know, company development? You've done that. And so I did a course for Harvard undergrads, which was, you know, how do you take an idea? And I made them come up with their own ideas of new sort of medicines that they would want to take into a biotech. So I had learned that, but in the first lecture that I always gave of this class, I would always say, well, the first time I started a company, which was Moderna, I knew less than a block of wood knows about starting a biotech company, because it was true. I had not done that before. And so I had zero skill set actually at it. Why should I have a skill set at it? I've never done it before. So I learned a lot during the formation of Moderna. It certainly helped that it was so successful, and it was very successful even from the very inception. It raised a lot of money. It made big pharma deals. It was very exciting, and I learned a lot. And I had always been sort of interested in science that has the potential to be translational. You know, science for science sake is great as well. You can study something your whole life, or many things that are wonderful to know about, discovering the unknown. And I did a lot of that as well. But I thought I was always interested in is if we make a discovery, is there any way we could envision moving that into, you know, patient health as an application for patient health? And I really got, after the foundation of Moderna, pretty much all the projects in my lab were focused on issues of, hey, if we achieve this goal, you know, this is an unmet need, an unmet technology or whatever. If we achieve the goal, could we potentially move this towards patients? That became sort of my driving force. And we made some discoveries in CRISPR. You know, we did not discovered CRISPR by any stretch of the imagination, but we used it very, very early, and we took it into blood forming stem cells. And we, you know, I think we were the first to put it into blood forming stem cells. And we targeted CCR5 with an eye on potential, you know, transplantation for HIV patients, because we knew from the Berlin patient that one person who's ever been cured of HIV had been transplanted by blood stem cells that were lacking this receptor, and just various other projects that, you know, indeed, when we achieved the question, it was like, okay, this is what we wanted. We were aiming at doing this, and now this can move off into a company startup. And now I knew more than a block of wood, so I knew how to do that. You know, gather the and I like I like the process of thinking early about what's the sort of therapeutic model, what's the business model, what people do we need around the table, what do we need to focus on, what diseases should we be thinking about. It's quite exciting, and I still do it to this day. I can't say no to good science. When there's really great science, it's really just, it gets my attention. I work on it. It's just the way it is. Science is inspiring. So I founded, Intellia Therapeutics, one of the CRISPR companies, co-founded with others that were more sort of fundamental CRISPR people. Jennifer Doudna being one. She won the Nobel Prize recently for discovery of CRISPR. Magenta Therapeutics, which is actually really my training as a blood stem cell biologist, with some studies that we showed for conditioning patients without chemotherapy and toxins before transplant using antibody-mediated clearance, and that worked really well. So the company was founded on that, and some other ventures as well. But it's addictive. It's fun. You get to work with smart people, of course, and with the goal of, if you're successful in that endeavor, you could impact. I've got 3 daughters, and I'm lucky that they're healthy young people. But I would imagine that if they were not healthy and they had some issue with them, that I'd be up day and night hoping that somebody was working on something that could, impact their health in a positive way. And all families feel that way about their loved ones when their loved ones get sick. So, to me, that's the motivating force for keep doing this. There are there are going to be people at the end of this that if you're successful, it really just totally changes their quality of life.
Yeah. Well, it must be very satisfying to see some of these things actually go from the lab bench into people's arms, as they're saying right now.
Oh, it's exciting. Even when the Pfizer vaccine was administered the other day, 2 days ago in the US, it uses the modified mRNA technology as well, same technology. So I was pretty satisfied to see that. I mean, even when, the early clinical trials for some of the other modalities, when it was going into patients and reading out positively and safely, obviously is a big deal. It was very exciting for me. But, who knew that a pandemic was going to be the first sort of application that should grab the attention of the planet? But it certainly has, because this pandemic that we're in is just so devastating and so pervasive to the lives of every single person on the planet.
So you said actually early on that the business model for vaccines is not good, and that's absolutely true. I've heard that several times. You can't, generally speaking, companies don't make vaccines for diseases that haven't happened yet, and it's very difficult. Except that's why flu vaccine is okay, because you have to keep making it every year. So there's a kind of business model there.
Yep.
But it is a challenge, and also, when we had the SARS outbreak, which hit Toronto badly, people were busy making vaccines, but then the virus went away and the vaccine went away, and the companies haven't made any money, right? And there's no point in them doing that. I worry, and I'm going to ask you the question, is, okay, we make this vaccine for this pandemic. Are we able to, well, maybe I know the answer to my question. The messenger RNA approach would be the most rapid way to respond to the next pandemic, right? So we can easily, we know it's safe, we know it worked. Next time, presumably, if Moderna has the sequence of, whatever the virus is, in 2 weeks, they just make the vaccine, and with a small safety trial, you could go. Is that going to be do you think that's going to be a feasible model for the future? So we stop pandemics before they get spread?
Well, you would, as a scientist, you can reason that very well, and maybe even many people could understand that right now. Though before a pandemic really it doesn't, not even a pandemic when it first starts, and it has to reach a certain phase before it. But if you were to start doing that prior to devastation really being revealed, there would just not be buy-in. There
No.
wouldn't be buy-in from the public. There wouldn't be buy-in from regulators. There wouldn't be, because ultimately you're vaccinating healthy people. And, why give a healthy person a medicine?
Well, we vaccinate healthy people against measles and mumps and everything else.
Well, it's a great point. And vaccines are about one of the longest, the medicines that work and are safe and effective. Yeah, I think buy-in is really the issue. We have short memories as humans. We make the same mistakes over and over again. I mean, it's we're smart, but we're incredibly stupid as well. So, in 10 years' time, we will have forgotten about this year, 2020. Honestly, it's hard to imagine, but we will have forgotten about it, and something will come again, and we'll be like, what do we do? I mean, it's incredible to me.
Yeah.
You would hope that this one maybe has created such a awareness that won't be true, but well, maybe I have too little faith in humanity.
I don't necessarily disagree. I do think there are many lessons we have to learn coming out of this pandemic, and we must learn them and not forget them. We have to remember how dreadfully unequal the effect of this pandemic has been on vulnerable and racialized communities, and we have to make sure that doesn't happen again. And that means not addressing anything about disease. It means addressing the unequal aspects of society. So this this should be a learning moment. Let's hope it is. But
yeah, well, the one thing
I'm not sure it will be, but I think, Well,
nations like Canada, I mean, Canada, is rational people. I'm proud Canadian, good policy making. I live in the US right now, so that's a funny place. The one thing that could be said at least, and part of the issue with this pandemic is the supply chain, basically. Moderna has said that it will make 1 billion vaccines by the end of next year. Pfizer has said also 1 billion. They're 2-dose vaccines. So that's 2 billion doses, but that's only 1 billion people vaccinated. I don't know what last count the population of the planet was, but what is it? Something like 8 billion or 9 billion, something like that. So a huge shortfall. There's other vaccines that are coming in phase 3. They're, by the way, adenovirus. They're almost all adenovirus based. That's also a pretty untested technology. I think that there's one approved adenovirus vaccine. It's for TB, tuberculosis. I don't think that, maybe there's another, but it's not a super well, off the shelf technology. It works though, and it's great. And I'm cheering on all those companies because we need, many more than 1 billion people vaccinated on the planet. We need 5 billion people vaccinated on the planet. So, and even the manufacturing infrastructure, both for mRNA and these large scale adeno, it just doesn't exist. So I'm hoping that countries, and I'd love to advocate Canada, and I have a little bit indirectly through our old colleague, Alan Bernstein, actually. I've been talking with him quite a bit about this, about having Canada invest in the manufacturing capability to be able to, because even if it's not for the next pandemic, adenoviral therapies are coming, mRNA therapies are coming. These manufacturing facilities can be used for that. And it'd be good for the economy. They're good, high paying technical jobs. And these medicines are coming. So why not invest in the infrastructure so that the next time, for example, when a global pandemic, back to the topic we were talking about, hits, we can maybe, without going through, long phase 1, 2, and 3 trials, maybe not yet, but maybe a big phase 1 study to show efficacy and safety, that would be sufficient to approve it and get it out much quicker and get the manufacturing behind it to have billions of doses as needed. So this is something that we could actually really take steps towards doing, and we should.
Well, I think your message and other people's messages through Alan and through the COVID vaccine task force in Canada, so that's been a strong message that manufacturing, it's not so much that people say, oh, Canada has lagged behind. As you say, the trouble is these are new technologies. You need special manufacturing setups to do this, but they are going to be something that's going to be needed in many different aspects. So I think you're going to see a push in Canada and certainly elsewhere in the world on that vaccine manufacturing side. So it's good.
Yep, it's good.
All right, we're coming towards the end. I've got a couple of little questions. How have you been surviving the pandemic?
Well, I mean, let's put it this way. I have it a lot better than most. I had a job and have a job that is not impacted by the pandemic, unlike many people on the planet. Service industry, and there's so many, the economic devastation of this thing is massive. So I was spared that. We've also stayed healthy. So I'm a biologist, and biologists are pretty good at, following the rules on, mask wearing and social distancing and this and that. Well, touch wood, it's not over yet. We're going through some dark months, but at least my family and my sister's kids, they're clinicians actually, and they got, one is, ENT. An ENT got COVID. What a surprise. Ear, nose, throat. What does he work with? He works with, the perfect place to catch COVID.
Yeah.
Luckily, they're young, healthy people. So generally speaking, those nearest and dearest to me have managed to stay healthy. When the pandemic first hit, we have a summer cottage in New Hampshire, which is really isolated in the country. And as soon as the kids were going to Zoom school, we thought, well, we could do Zoom school in New Hampshire. So we drove 2 hours from Massachusetts to New Hampshire, and we lived in our little, wood cabin for 6 months, actually. Okay. Yeah, so it was really, again, tough life.
Yeah, yes, we are lucky to be able to do it.
Totally lucky, yeah. So living it.
Wi-Fi connection that you're isolated.
I had to have a good one put in there because it was not possible to. I'd have to, when I'd take a call, I'd have to go outside the house onto a hill to get enough reception. I'd constantly be dropping calls. Okay, real first world problems I'm having here. I'm not complaining. Though when school started, we moved back to Massachusetts for the kids to go to school, and COVID puppy, like many others, got a COVID puppy, and he's a new little family member that we all love, and gets us out walking him every day, which is good. But I have to say, I'm going stir crazy. I'm going a little bit bananas being in the house for so many hours every day for so long. I'm looking forward to traveling again and interacting with people out.
As you say, we are very lucky. We're here, we're healthy, we have jobs, and what a lot of what we do can be easily done virtually, and it's not the same, obviously, as in person, but we have a lot of opportunities. So.
Pretty easy.
And I've been able, through the Gairdner Foundation this year, to have a number of great online virtual conversations and webinars and meeting people and interacting with people all over the world and being able to put out content all over the world better than we ever do in person.
Yeah, that's fantastic.
There are definitely positives to some of these.
Oh, it's amazing, actually. Again, we just a few moments ago talked about how stupid people can be sometimes, but they're pretty innovative too. And the innovations that have come in this year, we respond to adversity, we adapt, and we come up with things. Humans are pretty clever and industrious in that regard. So I've been pretty impressed to see, young people and just people around the world sort of adapt and come up with new business models and totally new industries. And of course, some industries have been suffered terribly. And but others are adapting. It's pretty cool to see.
Yeah. Well, Derrick, it's been a real pleasure to talk to you today. And as I say, through the aegis of Zoom, we've got together. We'll have to get together in person sometime again soon. It's been really a pleasure to talk to you. And thank you so much for giving us a bit of the history of Moderna, a bit of your history. And you're welcome back in Canada anytime, you know.
Yeah, thank you, Jen. It was really a lot of fun. Really enjoyed it, and great to see you again.